Skeletal and cardiac muscles, as well as liver, are major consumers of fatty acid for fuel. Many other tissues burn fat, for example, between meals, when blood sugar level is low. How is energy extracted from fatty acids? We are going to discuss fatty acid degradation, a process also called beta-oxidation. As you recall from a previous video, fatty acids are stored in adipocytes and mobilized by lipase enzymes. Fatty acid bound to proteins are transported from the lipid droplets to the bloodstream. In the bloodstream, fatty acid bound to serum albumin are transported to tissues. In tissues, fatty acids are taken up by transport proteins and must be delivered to mitochondria, where they are metabolized. How do fatty acids enter the mitochondria? Fatty acids, such as palmitate, cannot diffuse freely across the mitochondrial membranes. The acyl-carnitine shuttle modifies fatty acids, allowing their movement through the outer and inner mitochondrial membranes. The acyl coenzyme A synthase, found at the outer mitochondrial membrane, attaches coenzyme A to palmitate, forming palmitoyl coenzyme A. This reaction requires the hydrolysis of ATP, leading to the production of AMP. Note that ATP is converted to AMP, not ADP. So it takes 2 ATP to regenerate 1 molecule of ATP from AMP. Therefore, we say that the formation of palmitoyl coenzyme A requires 2, not 1 ATP molecules. This will be taken into account later when we calculate the amount of energy delivered from palmitate oxidation. The next step in the process is the replacement of coenzyme A by carnitine, forming palmitoyl carnitine. This reaction is catalyzed by the carnitine palmitoyl transferase, or CPT1. CPT1 is located in the outer membrane of the mitochondria and is tightly regulated. CPT1 acts as a gatekeeper for fatty acid entry into the mitochondria. Palmitoyl carnitine is transported in the intermembrane space via the transporter called porin. Alternatively, palmitoyl coenzyme A could be transported first and then converted to palmitoyl carnitine inside the intermembrane space. The carnitine moiety allows the fatty acid to be shuttled across the inner mitochondrial membrane. This process is made possible by a translocase. Now, the palmitoyl carnitine is inside the mitochondrial matrix. Before it can be subjected to beta-oxidation, the fatty acid has to be transferred back to a molecule of coenzyme A. CPT2, attached to the inner mitochondrial membrane, catalyzes the transfer. Carnitine is released, palmitoyl coenzyme A is formed and ready to be broken down and produce energy. So how is the acyl-carnitine shuttle regulated? As I said before, CPT1 is a major regulatory site. CPT1 is allosterically inhibited by malonyl coenzyme A. As you recall, malonyl coenzyme A is a building block of fatty acid synthesis. Here, malonyl coenzyme A inhibits fatty acid oxidation. It shows that fatty acid synthesis and beta-oxidation are reciprocally regulated by malonyl coenzyme A.
We saw in the last video that AMPK inhibits fatty acid synthesis. Does AMPK also affect beta-oxidation?
Yes, AMPK promotes beta-oxidation. Remember that AMPK phosphorylates and inhibits ACC when energy supplies are low. As a result, less malonyl coenzyme A is formed, meaning that fatty acid synthesis slows down. CPT1 is no longer inhibited, and beta-oxidation increases. This reciprocal regulation helps the cell to respond to a low-energy crisis. Once fatty acyl coenzyme A molecules have entered mitochondria, beta-oxidation can start.